Learning. Today we are going to discuss about the classification of monosaccharides. Already we discussed that monosaccharides are the single carbon containing compounds. And these are classified based on the number of carbon atoms present in them. And also they can be classified based on the presence of carbohydrate. The naturally occurring monosaccharides and that containing 3 to 7 carbon atoms per molecule. And whenever we are denoting the carbohydrates and that are containing the suffix term as the roots, and that can be classified depending upon the number of carbon atoms and that are of the based on that here these are considered as the trioses, tetroses and pentoses. Triose means 3 carbon atom containing compounds, whereas the tetroses are the 4 carbon containing compound, pentoses are the 5 carbon containing compound. Whereas hexoses are the 6 carbon containing compound. And here, and these monosaccharides, these are mainly considered, these are also classified depending upon the functional group as the aldoses or the ketones. Aldoses are the sugars that containing aldehydes as the functional group, whereas the ketoses that containing ketone as the functional group. And combining these classification system, and that means that is number of carbon atoms and also number of the what is the functional group that is present, and that can be classified as the aldotetrosis or aldohexoses, or we can say depending upon the number of carbon atoms here. That is aldotetrosis, aldopentoses, and if it is functional group is ketose, then ketopentoses, ketohexoses. And this glucose and fructose, and that are the main examples of the aldohexose and ketohexoses. And these can be, these are the some of the carbon atoms containing compounds, and that are containing 3 carbon atoms, 4 carbon atoms, 5 carbon atoms, and 6 carbon, 7 carbon, which can be named as in terms of the functional group as allos, which are glycerols, and this is also termed as glyceraldehyde phosphate, glyceraldehyde, and this is dihydroxyacetone, and next tetroses are called as the erythrosis, erythrolysis, if the functional group is ketone. Whereas pentoses that are named as the ribose, and if it is ketose group, functional group is present, that is called as the residue. Okay, and depending upon that here, these can be classified, and we also discussed the properties of the trioses and tetroses like that here. And here, coming to the main point, that are the properties of the this monosaccharides. Here, first of all, before going to the properties, the monosaccharides. The monosaccharides structure, and that is existing in the form of 2 conformation. One is open chain conformation, or that is called as the straight chain conformation, and the other one is called as the cyclical structure. Here, the open chain conformation, and that means here the 6 carbon atoms of the glucose that are arranged in a straight line, and that is called the open chain structure. Because here the 2 ends, that means the terminal and open end, that are remaining separate. These are not linked structure, and that are called as the open chain structure. And this structure was proposed by Fitting and Bayer. And this structure also proposed by Fisher's projection formula. And this is the main one that we are using in representing the structure. That is called as the Fisher's projection formula. And here, when we take the structure of glucose here, it contains the molecular formula of C6H12O6. And in straight chain conformation, that is in Fittig-Bayer conformation, the structure is written as here. And whereas the straight chain structure, and this is written as here. Okay, actually in your questions, in your examination, no structures will ask you. Sometimes 1 or 2 questions will ask you from the structure, but these are mainly based on the theoretical points. Okay, next coming to the cyclical ring structure, and this cyclical ring structure, and that was proposed by Haber. and that is called as the Haworth projection formula. Haworth's projection formula. According to the Haworth's, the sugars are existing in mainly two types of the ring. One is called as the furanose ring, which is the 5-membered ring. This is called as the furanose. And that means here, this is containing 4 carbon atoms linked with the oxygen. That is called as the furanose ring. Whereas the pyranose ring, and that is existing as a 6-membered ring. 6-membered ring, and that contains 5 carbon atoms linked with the oxygen. And what are the properties means already we discussed some of the properties that are colorless structure, crystalline, and these can be soluble in water and that can be tasted as the sweet. Okay, and here depending upon the optical activity, here these are in the form of two forms. One is dextrorotatory form and the other one is the levorotatory form. Dextrorotatory form means that is directed towards to the right direction. It is directed towards to the right direction, whereas the levorotatory means that is directed towards the left direction. Whereas the mutarotation, the change in specific rotation of an optical active compound is called as the mutarotation. And during the mutarotation, here mainly 3 states are possible. One is the equilibrium state, and that is converting into the beta D glucose conformation, and it also be converted into the alpha glucose conformation. And alpha glucose conformation having the specific optical rotation of the plus 112.112 degrees, whereas this equilibrium state is present at the 52.5, and whereas this is present at the 190. Okay. And next coming to the glucosides. Glucosides. Actually, we know that the sugars are mainly connected by bond that is called as a glycoside. And here, whenever the glucose, the monosaccharide, it is reacting with the methyl alcohol means that is CH3OH. When it is reacting with the methyl alcohol means here the primary carbon atom and that is linked with this methyl alcohol, and that results in the formation of the glucosides. In examination, the question will arise like this. Monosaccharides reacts with the alcohols, resulting in the formation of glucosides. Coming to the esterification. Esterification means here the glucose molecules when reacting with the anhydrates, and these are resulting in the formation of esters. And reducing agent. These monosaccharides are acting as the oxidizing agents such as hydrogen peroxide. And in this here, what is happening is the sugars are mainly oxidized. So the oxidizing agent is becoming as a reduced. Next, formation of oxygen. Here, the monosaccharides are mainly resulting in the formation of the oxygens when it is reacting with the phenyl hydrogen reaction. Okay, coming to the disaccharides. Disaccharides are the consisting of 2 sugars that are mainly joined by glycosides. And here the disaccharides, and these are also classified into 2 types that are depending upon the name, what are the units that are present here. And these can be classified into homodisaccharides and heterodisaccharides. Homodisaccharide means anytime homo means it is representing as the similar, hetero means different. Here, and if it is containing the same units means that is called as the homodisaccharide. If it is containing different units means that is called as the heterodisaccharide. And what are the homodisaccharides means here? The maltose is consisting of the 2 glucose units. Here you have to remember what is the bond that is formed. That is alpha here. And here that means this is containing alpha 1,4-glycosidic. 1,4-glycosidic bond. Okay. And here the maltose is the homodisaccharide that is mainly containing 2 glucose units. 2 glucose units which are linked with the alpha 1,4-glycosidic. Okay. Whereas the isomaltose here and glucose, that means it is also containing of the glucose units. It is also containing of the glucose units which are linked by alpha-1,6 glycosidic. Whereas the cellobiose is mainly consisting of the beta-D glucosides, and that consisting of the beta-1,4 glucosides. And here, the anomeric carbon is free here. That means it is not reacting in the reaction. Only the terminal one that is reacting here. And that means the anomeric carbon that is free here. And whereas the reducing property, it has the activity of reducing. And also these disaccharides that can be produced by the hydrolysis of the polysaccharides. Here, and this starch hydrolysis by the action of amylase and amylopectins, and that resulting in the formation of these maltose units. Maltose units. Actually, here the starch is made up of glucose units, which is consisting of the alpha-1,4 bonds and 1,6 bonds also. And that results in the hydrolysis, resulting in the formation of the alpha-1,4 means that is resulting in the maltose units. Okay, whereas the dextran resulting in the formation of the isomaltose. Whereas the cellulose means it also contains the units of the beta-D glucose. And that means the cellulose on hydrolysis resulting in the formation of this cellulose. Okay, and that means here starch resulting in the formation of maltose and dextran resulting in the formation of isomaltose, whereas the cellulose resulting in the formation of the cellulose. And these are the structures of the maltose, isomaltose. Here you can observe here. What are the bonds set up with? Here, and this is the glucose units. These two are the alpha glucose units. These two are the alpha glucose units. And these two alpha glucose units are linked up by here alpha-1,4 glycosidic. That means here in the first glucose, the first carbon is involved, whereas in the second glucose here the fourth carbon is involved, and that resulting in the formation of the maltose. And like that here the isomalt is also formed. You have to remember the main units and also the bonds. And next coming to the heterodisaccharides containing also of many units of the heterodisaccharides containing different units of the monosaccharides. And that means here these are mainly formed of the two different monosaccharide units. And for this, the main examples are the sucrose and lactose. And sucrose mainly consisting of the alpha-D glucose and beta-D fructose, which are linked by alpha-1 beta-D glucose, alpha-1 beta-2 glycosidic bond. Okay, whereas lactose and that containing the beta-D galactose, that means the first unit is the galactose and the second unit is the glucose. But galactose and glucose that are linked by beta-1,4 galactose. Okay, whereas here in this sucrose here, and this is the main important bit now. And here in this here low free aldehyde or ketone group is presenting sucrose. And that means here what is the bond that is formed? Alpha-1 beta-2. Beta-2 means what is the one? Fructose. And alpha-1 means this is the glucose. That means here the first carbon and the first unit that is containing glucose and that containing the aldehyde group, and beta-2 means that containing the ketone. And that means here no free aldehyde of ketone group. That means the anomeric carbon atom that are containing no free aldehyde of ketone group. Whereas in lactose here, the free groups are present here. Okay, and here if it is containing free groups means that is called as the reducing sugar, and if it is not containing any free groups means that is called as a non-reducing sugar. That means here we have to remember one thing now, but that is specific is sucrose is called as a non-reducing sugar. Sucrose is called as a non-reducing sugar because of no free aldehyde or ketone. No free aldehyde or ketone. Okay, and what are the hydrolysis means? Here, the sucrose mean mainly consisting of the glucose and fructose. That means here, and this can be hydrolyzed by the enzyme that is called as the sucrose or invertase enzyme. Okay, here the main enzyme that is acting on sucrose is called as the invertase enzyme, which is resulting in the formation of the glucose and fructose. Okay, whereas here and this lactose that can be hydrolyzed by the intestinal lactase enzyme, which resulting in the formation of the galactose and glucose. Okay, and this sucrose, and this can also be called as a table sugar. It is also called as a table sugar, cane sugar, or wheat sugar. Whereas here lactose that is mainly present in the milk, so it is called as the milk sugar. And also it may appear in during late pregnancy and during lactation. Lactation individuals, women mainly containing this sugar, that is lactose sugar. Okay, and that means here in the disaccharides, we have to remember the main points of what are the linkages that are present and what are the homodisaccharides and heterodisaccharides. Here, what are the disaccharides that we here that discuss maltose, maltose, isomaltose, cellulose, which are the homodisaccharides. Whereas hetero means lactose and sucrose. These are the hetero. Okay, and these are the homo. Okay, you can also write the bonds here. What are the units that are present? And also you can note down the bonds that can easily remember. Okay, next come to the polysaccharides. What are polysaccharides? Polysaccharides are the sugar that are comprising of the that are releasing or hydrolysis of the monosaccharides. Okay, and that means these are containing many number of the monosaccharide units. So that was called as the polysaccharide. Poly means many. And here the polysaccharides, these are not called as the reducing sugar. Actually, the remaining sugars are called as the reducing sugar, whereas the sucrose is called as the non-reducing sugar. And here the polysaccharides are also the not reducing sugar, because here the anomeric carbon atom, the functional group containing carbon atom that is involving in the linkage, glycosidic linkage. So here the polysaccharides are called as the non-reducing sugar. And these can be classified into homopolysaccharides and heteropolysaccharides. Homopolysaccharides are the polysaccharides that are made up of one type of the monosaccharide unit. Whereas the heteropolysaccharides are the polysaccharides that contain more than one type of the monosaccharide unit. Okay, and for this, the main examples coming to the homopolysaccharides here, the starch is the main polysaccharide. Already we discussed it. Here, the starch is mainly consisting of the D-glucose and these are insoluble in water because of the high molecular weight. And these are mainly forming the colloidal suspensions with the water. So here, the starch and that is insoluble in water. Whereas the starch is also considered as a storage compound, and because of it is made up of the glucose units. Okay, and it is mainly linked with the amylose chains and amylopectin chain. And what are these means here? The amylose means these are the straight chain structures that are formed by alpha 1,4. Here you can see these are the straight chain linkages, and that are mainly formed with the alpha 1,4 glycosidic. Whereas the amylopectin here, and these are the branched ones. These are the branched structure that can link with the 1 to 6 bond. Here, this is the first bond, and this is the sixth one. Okay, and that means here amylase is mainly formed with the alpha 1,4 linkage, whereas the amylopectin that is formed with the branches that is connected by linkage called as the alpha 1,6. Okay, and here, as coming to the structure of amylose, here the amylose that is mainly forming as a helix-like structure. It also causes the blue color chain on reaction with iodine. This is the main significance because it can identify the starch. Here, iodine reaction with the starch will result in the formation of the blue colored gold. Blue colored gold. Okay, and it is also insoluble in water, wholly soluble in water. Okay, whereas the amylopectin actually it is forming some like violet color chain, red violet color chain on reaction with iodine. Okay, and next coming to the glycogen. Here, one example is the starch, that is the homopolysaccharide, and that also consisting of the alpha glucose unit. Okay, whereas the glycogen, this is also a storage one, and this also consumed, it is mainly present in animal sugar. Starch is present in plant cells. Okay, and here the glycogen, that is the main reserve of the animals, and that is mainly reserved in liver and muscle. Okay, and here this glycogen is stored energy for the organism, and it is similar to structure of amylopectin. Okay, and here depending upon the number of branches, these are different. And this is the glycogen mainly gives the red violet color with iodine. It is forming the red violet color with iodine. Coming to the cellulose here, the B glucose molecules here, the starch and glycogen. And these are also consisting of the glucose unit. And here these are mainly consisting of the alpha one. Whereas here the different one, that is the cellulose here, that is mainly consisting of the beta 1,4 linkage. And these beta 1,4 glycosidic linkages, and that are linked with each other, and that forming the 100 to 1000 units long structure. And here, and the hydrogen bonds are also formed in between them, that resulting in the formation of microfibrils, and these microfibrils, and that are forming the compounds hemicelluloses, and this hemicelluloses and calcium, and that resulting in the formation of the cell wall. Here you can see here the cellulose means it is mainly consisting of the beta 1,4 glucosidic acids. Okay, and that resulting in the formation, that means these are consisting of the glucose units of beta 1,4, and these are resulting in the formation of the microfibrils, and these microfibrils are forming thicker bundles. Okay, and that are grouped together by other compounds such as hemicellulose. These are linked with the hemicellulose, and these hemicelluloses and also linked with the peptide form, and that is of the calcium peptide, and that resulting in the formation of the plant cell wall. Okay, and because of the beta linkage, cellulose has a different shape to the amylose, forming the extended side chains with hydrogen bond. Because of the presence of the hydrogen bond, it is mainly occurring as a rigid structure. Okay, and also it is an important structural polysaccharides, and it is mainly provided in the plant cell walls, which is providing strength and rigidity. Okay, and most animals do not have the enzymes to digest the cellulose. Only plants have the ruminants have the enzymes, and that cellulose enzyme, and that helps to break down the cellulose. Okay, and this cellulose is also important in industrial area because it is used in wood, paper, cotton, cellophane, and these industries. Okay, next coming to the chitin. Chitin is a polymer, and that is mainly made up of glucosamine units. Glucosamine. Here you can observe N-acetyl glucosamine. These are the units of chitin, N-acetyl glucosamine. Okay, what are the functions of chitin means here? And this is mainly found in shells of beetles to webs of spiders. Okay, it is present in all around us in plant and animal creation. And also this chitin, that is the fungal cell wall. This is the most important point now. Many times this question asked, the fungal cell wall is made up of chitin. And sometimes they will ask like units, N-acetyl glucosamine units. Okay, and it is also considered to be a spin-off cellulose because here these are mostly similar here. Okay, and cellulose contains a hydroxyl group, whereas the chitin contains the acetamide group. The chitin contains the group called is the acetamide. And chitin is existing as a natural polymer, and that means it is existing as the combination of the different elements that are existing on the end. So this is called as a natural polymer. Okay, and chitin is a very thin material, and that is help to protect the insect against harmful ones. And next coming to the inulin. Inulin is present in the roots of the dandelion, and that means it is mainly present in the tubers and roots. Tubers and okay, and here and this inulin is called as the polymer of fructose. It is called as the polymer of fructose. And it is formed in the plants by eliminating a molecule of water from the glycosidic group of carbon atom of beta D fructose unit. And that means here the functional group unit, and that can be removed by using it. Okay. And next coming to the peptides, peptide compounds. These are also the cellular compounds that are mainly forming the cell wall. And it is a polysaccharide of alpha D galactorinic acid. Alpha D galactorinic acid, because of this here these are called as the polygalacturonoids. And coming to the mucopolysaccharides, and these are composed not only of mixture of simple sugar, and that means here, and these are mainly containing, these are present in the form of amino sugars. Amino sugars. Okay, and next coming to the hyaluronic acid. And hyaluronic acid, and which is called as the lubricant, okay, and that is mainly present in our eyes. And it is the most, this is the heteropolysaccharide. This is called as the heteropolysaccharide. Because of the presence of the D-glucuronic acid and N-acetyl D-glucosamine units. D-glucuronic acid and acetyl glucosamine units, and that are linked by beta 1, 2, 3, 2 and 1, 2, 4. Okay, here the main linkages are beta 1, 2, 3 and beta 1, 2, 4. And here the main units are D-glucuronic acid and N-acetyl D-glucosamine. And it's coming to the chondroitin. And chondroitin, it is mainly present in cartilaginous tissues. Cartilaginous tissue. And it is present in the form of mucopolysaccharides, chondroitin sulfates. And this is also similar to the structure of hyaluronic acid. Okay, and this also containing beta D bonds, and this is mainly contained in place of the glucuronic acid. It is mainly containing the galactosamine. Galactosamine. Okay, that means here, and this is also a unit that is consisting of the units of galactosamine rather than the glucosamine. And it is called as the beta D glucuronido 1,3N acetyl D galactosamine. Okay, and that means here the chondroitin mainly containing the units of D glucuronic acid plus N-acetyl D-glucosamine, which are linked by beta 1,4 linkage. Okay, and these are mainly present as the structural components of the cartilages and tendons and also bones. Okay, and it is also present. That means here the what are the linkages that are present here of beta 1, 2, 3 and 1, 2, 4. Okay, and here these are the main heteropolysaccharides that are present here, and that are the hyaluronic acid, dermatan sulfate, chondroitin sulfate, and in chondroitin 4 sulfate is there, 6 sulfate is there. And it's coming to the dermatan sulfate here. The dermatan sulfate, it is a mucopolysaccharide which is similar to the chondroitin, and here the glucuronic acid, and that is replaced by L-iduronic acid. What are the linkages means 1, 2, 3 linkage and 1, 2, 4, and that is of the beta linkage. Okay. And next coming to the keratosis sulfate here, and also you have to remember chondroitin sulfate B is similar. It is called as a dermatan sulfate. It is called as a dermatan sulfate. Okay, next coming the keratosulfate, and this keratosulfate here the uronic acid component is replaced by D-galactosphere. D-galactosphere. Okay, and here the linkages are beta 123 and beta 123. And next, heparin. Heparin consisting of the D-glucuronic acid units. Okay, and this is mainly acting as anticoagulant, and that means it is mainly preventing the coagulation of the blood by prohibiting the prothrombin thrombin conversion. Okay, and these are the main polysaccharide units, and that are consisting of the different units and what are their occurrence and what are their function. Okay, starch is called as a polymer of glucose, glycogen polymer of glucose, cellulose is polymer of beta D glucose, beta glucose, whereas insulin is a polymer of fructose, whereas pectin means that is a derivative of galactose. Okay, hemicellulose means that consisting of the pentoses and sugar unit. Lignin, and that is polymer of glucose. Chitin is a polymer of glucose. Okay, and murine is linked. It is a polysaccharide that is cross-linked with the amino acid. And hyaluronic acid, it is a polymer of sugar acid. Okay, chondroitin and heparin, and these are all related ones. Okay, and these are the some of the polysaccharide and their functions. Okay, here what are the functions means here? Starch, and that is mainly the plant storage food, whereas glycogen, storage of preserved food in animals, cellulose present in cell walls, inulin, and that is the reserve food, pectin, it is present in cell wall, hemicellulose, it is also the cell wall matrix, lignin, and it is also the component of cell wall, chitin, it is mainly as the fungal cell wall and also exoskeleton, murine, which is a structural protection, hyaluronic acid, it is a ground substance which is protecting, and chondroitin presenting mainly connective tissues and matrices, and heparin, which is acting as an anticoagulant, and gums and mucilages, which are retaining water in dry season. Okay, and this is all about the polysaccharides and disaccharides. Okay, we have to frame a table and what are the main units that are present in the structure and what are the main bonds and their function. And like this, you have to frame with some table for disaccharides and polysaccharides. Thank you.